Hi everyone. As Louis said, I'm Chris Michaels. I manage the herpetology section at ZSL London Zoo, and I'm going to talk to everyone today about managing water quality for amphibians. And I want to just start with the caveat that, as everyone knows, amphibians are extremely diverse animals and also extremely sensitive ones. So this is intended to be a broad brush approach to the different principles to consider when managing water quality, as opposed to giving a step-by-step guide to managing any particular system. So I'm more than happy to, if people want to get in touch afterwards, if they've got a particular question about a species they manage or are interested in managing, whereas this is supposed to give the principles of how to apply the knowledge to any particular context. Also, if you hear frogs in the background, it is frogs, and I apologise if they interrupt too much. I can't really tell them to be quiet though. So without further ado, I'll get on with it. So yeah, I want to talk about two main things in this presentation. It's going to be water quality and water chemistry, which are linked terms, but water quality is essentially the result of biotic and abiotic factors in an aquarium and of their interactions, whereas water chemistry is just the basic chemical characteristics of water in a particular environment, as opposed to how the organisms living in it affect it. And I'm going to look first of all through this talk at the really important parameters that we need to be thinking about when we're managing aquatic amphibians, and then secondly look how we control those factors, especially water quality. So I'm going to start with probably the most important thing to know about when you're managing aquatic amphibians, and this is the nitrogen cycle and how it relates to nitrogenous waste. So any animal which is living in an aquarium is going to be excreting animal waste, and it's also going to be not eating all of its food, etc. Things are going to die or be added dead into aquaria. And those things, as well as animal waste, can be broken down by bacteria. And the end result of those interactions is the three chemicals that we refer to as nitrogenous waste. They are ammonia and ammonium, nitrite and nitrate, which you can see here in this red ellipse. And the most critical part of the nitrogen cycle is the conversion of ammonia to nitrite to nitrate through bacterial action, as well as the uptake of those chemicals by plants. So we're going to touch back on that several times, so just keep it in your head. So why are these chemicals so important? Basically, they're toxic. They are excreted from the body because if the animal didn't do that, it would poison itself. But in an aquarium, they can accumulate to levels which can be just as toxic. So the three chemicals are in reduced toxicity relative to each other. So ammonia is the most toxic. The precise toxicity levels really depend on the species, but relatively trace amounts can cause quite a lot of damage to amphibians. And this basically mainly causes tissue damage. You can see in this axolotl here, or what's left of an axolotl, how the gill filaments have been all but erased by the ammonia, and the entire skin is damaged and reddened and irritated by the ammonia. And the other thing that ammonia will do is it will displace dissolved oxygen from the water. So in this case of this axolotl, not only does it have a reduced respiratory surface because it's lost its gills and it's got damaged skin, there's also less oxygen in the water for it to uptake. Nitrite, so the middle one, is about 10 times less toxic than ammonia. And this has some very different but equally toxic effects. It has a really high affinity for hemoglobin, so the chemical in the blood that carries oxygen, and it will bind irrevocably to it so that that molecule can never carry oxygen again. And you can see on this slide what happens to the blood. On the left, normal blood, and on the right, blood that has been exposed to nitrite, and it's developed brown blood disease because of the colour, more properly called methemoglobinemia. And the animal will essentially eventually asphyxiate because none of its blood can carry oxygen again. And nitrate is 10 times less toxic again, and this really is something to worry about in the longer term. It can cause damage to things like kidneys and other tissues. And all of these chemicals will also, if they're in unnaturally high amounts, will cause stress to the animals. It will depress immune function due to that stress, and it will also trigger metamorphosis in some larval amphibians. So as well as potentially killing animals in a short term, long-term exposure to sublethal levels can also have really bad effects. So we need to make sure we're managing these compounds, and I'll come to how we do that in the second part of the talk. Important parameter to think about is pH. This is just a measure of acidity or hydrogen ions and alkalinity, or the number of hydroxyl ions in the water. Most amphibians are adapted to a particular range of pH linked to the places that they're found in the wild, and individual animals will also physiologically adapt to the environment in which they are raised as well. So you've got a combination of evolutionary adaptation and individual physiological adaptation. And keeping animals outside their habitual range can be stressful, and extremes outside the range can cause tissue damage, so chemical burns again. They can affect osmotic function across the skin and the gills, and they can also affect the exchange membranes in other ways. So you can cause lethal effects or sublethal effects by having improper pH. Hardness and alkalinity is another important parameter. So alkalinity is the capacity of water to buffer against a pH drop, so to absorb hydrogen ions and stop the pH from crashing. And it's mainly caused by dissolved minerals that are in the water, and this is often referred to as hardness. There are two types of hardness: general hardness, GH, which refers to all dissolved divalent metal ions. That is especially things like calcium and magnesium. They're divalent because they are 2+. They have 2 charge on them. And obviously calcium and magnesium are familiar because they're important as part of bones. And carbonate hardness is a subset of that, and it's specifically all dissolved carbonates, especially carbonates and bicarbonates. And these are really relevant to amphibians, because if amphibians have been adapted to a particular level of hardness in the environment, too much or an increase in mineral content of the water above that level can lead to mineralization of tissues, and reduction in hardness under the level that an animal is adapted to can result in things like metabolic bone disease, because amphibians are capable of extracting minerals from the water across the skin and especially across the gills for gilled individuals. And also hardness and alkalinity will influence pH. Minerals are typically increase the pH in water. So generally the harder the water, the higher the pH. So there's an interrelation there. Dissolved gases are also important. The most important two to think about are carbon dioxide and oxygen. And these are important for respiration of all organisms in the water, and also photosynthesis. So not only of the plants that, as we'll see shortly, are important for maintaining a healthy aquarium system in many cases, but also for some amphibians that have symbiotic photosynthetic algae within their cells, things like spotted salamander, egg masses. And they can also, apart from being directly important for the physiology of the animals, they can directly influence pH. So for example, carbon dioxide, when it dissolves in water, forms carboxylic acid, and that will depress pH. So when we're thinking about gases, we're not thinking just about animals breathing and photosynthesizing, but also the influence on the chemical environment. Tannins and dissolved organic carbon are basically humic compounds that come from decomposing and leaching organic matter, and these are the things that make an aquarium become what's often called as a black water aquarium, so where the water looks like black tea or whiskey or similar. And these compounds come out of especially vegetation. They're acidic, the humic acids and tannic acids, so they suppress pH again to give an alkalinity. So by adding tannins to a system, for example, the pH can be lowered and it can become even acidic, even though there is a mineral content there. They also have physiological effects, including making calcium biologically less available or unavailable. And that can be really important to consider, as well as the inhibition of some bacteria and fungi. And the picture you can see there is a Theloderma stellatum tadpole. And like many tree hole breeding frogs, these tadpoles develop in situations which are saturated with tannic acids that leach out of the trees that the rainwater collects in. And for these species that have adapted to essentially living in an antimicrobial, if you don't get the levels of tannins correct, you can have animals becoming susceptible to invasion by or environmental bacteria and fungi that can cause lethal infections. So as well as those main sort of bits and pieces in terms of things you would normally find in water, in a natural environment, we also need to consider contaminants and pollutants that, rather than having a level which you can get right for an animal, you need to just avoid entirely. So some really common things that you might have to deal with are include heavy metals, things like copper, lead, mercury, etc. These usually derive from heavy industry or potentially groundwater, and also old pipework, and they're cumulative toxins. So as an animal is exposed to it, they will accumulate these metals irreversibly within their tissues, and eventually these will start to have things like neurological impacts. So it's really important that we are treating water to avoid contamination with these, because once the damage is done, it's done, and we can't take it back out of animal systems. Insecticides may contaminate aquarium or other aquaculture water, either from agriculture or from domestic products, and very low levels can kill. They're also associated with developmental problems in amphibians, and they're things like pyrethrins and DDT, etc., etc. And depending on where you are in the world, these may be more or less of an issue. I know there are some places, for example, in the US, where some pesticides have got so high that people have reported sex biases in their animals because of developmental impacts. Fertilizers likewise may contaminate from agriculture, and again these can play havoc because they are based on nitrogenous waste products, ammonia and the like, and they can also influence things like algal blooms and plant growth inside enclosures that can affect the physical environment. And finally, cleaning chemicals, often overlooked, but in most amphibians facilities, one thing you will definitely find other than amphibians is cleaning chemicals for disinfecting water and surfaces, etc. These can also be extremely toxic to aquatic life and to the life support systems that we use to maintain appropriate conditions. So we need to also consider ways of stopping these things from contaminating our water. So I've covered the main parameters, the sort of usual suspects when we're doing with the basics of water quality, but there are loads of others, including things like phosphates, which can affect algal growth, micronutrients that are important for plant growth, total dissolved solids, which are things other than the different minerals we saw within the hardness section, dissolved in water, things like silicates, and for those few amphibians that don't live in fresh water, salinity and conductivity. But there's a whole host of other parameters, and the general message is to research your species that you're going to be working with, and if you find that there's a particular parameter which seems to be important for them, to look at ways of managing that. I can't go into them all here because I would be here forever. So we've looked at the basics of what sort of things we need to be thinking about, and I'm now going to move on to how we control water quality within a system. And I mentioned this before, but I'm going to say it again because it is the most important principle of managing water quality, is to know your species. There's a whole diverse range of different amphibian species, and each of those has adapted to its own ecological niche and has different optima in terms of water quality, and also different ranges of tolerance. So work out which species, what the needs of your species you're working with are, based on field data and also based on talking to other husbandry professionals, and also the specific background of the individuals involved, so how they've been managed historically, and use that to design and inform the management of water quality for your species. It's impossible to manage anything properly if you don't do appropriate monitoring, and water testing is absolutely critical to underpin anything in terms of water quality control. And this needs to be done frequently. Infrequent testing is almost pointless because you're only getting a snapshot of water quality at each time, and if you test infrequently, you're not going to pick up any patterns. It's really, really important to note that it's impossible to gauge most aspects of water quality by eye or even by giving it a sniff, because the quantities of the compounds involved that can have physiological effects on amphibians are so low that we just can't pick them up that way. So if an aquarium smells of ammonia, then the levels will be extremely high and way beyond the point where water testing would have discovered a problem. It's also important to record your water testing and keep those records in an orderly and systematic way. And that's important because you need to be able to look back at the history of a system in order to work out what's going on with it, and also to correlate that with changes in behavior and health in the animals you're keeping, and also any changes or interventions you make with regards to managing the water quality. So as a rule of thumb, I would recommend testing at least once weekly for established systems, i.e. ones which have been running without problem and where you're confident that they are stable. And then for any other system, either a new system where you've just set it up and you need to make sure that things are settled as you intended, or where you've had an issue, so you've detected the problem and tried to rectify it, daily tests should be conducted to make sure that you detect a problem in its early stages rather than leaving it too late. And I should also add that water tests should also be used if you detect anything strange in your animals. So if, for example, they try to leave the water, or you see physical symptoms like we saw in that axolotl or in the other slide, one of the responses to ill health for an aquatic amphibian should be water testing. How do we water test? There's oh so many choices available. There are 4 main different types of water testing equipment. Dip tests, where you just put almost like a pregnancy test type thing into water, a little coloured square changes colour. Liquid reagents and titration tests, which are basically still by eye, and you can buy these in most aquatic shops. And then specialist equipment, including probes and spectrophotometers. So these all have different degrees of utility. Dip tests are great because they're cheap, they're really easy to use. You just swim it in the water and have a look at it, and they do multi-parameters, so you can do a whole suite of tests with one thing. But I would strongly not recommend them unless you have absolutely no choice, because they are really inaccurate. The concentrations of chemicals in the pads changes, and they rely on the human eye, so there's also a human error in terms of judging the colour. So these can be okay for field work, but it's really better to use any other test if you can do. Liquid reagents and titration tests are still quite cheap comparatively. They're much more accurately if you use them correctly, because the concentrations of chemicals are better controlled. But they're a bit more complicated to use. You've got to add 3 drops of this, 2 scoops of that, shake for this length of time, etc. And that means there's more room for human error, both in terms of measuring out and following the instructions, and in terms of interpreting colour. And they're also not brilliant if the water in the system is tinted. So let's say you have an enclosure of Theloderma tadpoles and the water is already a yellowish brown colour. It's really hard to work out how red something is by eye while allowing for the reddish yellow water that was there to start with. So that can become a bit more difficult. Specialist probes are available for certain things like conductivity, dissolved oxygen, a whole bunch of other stuff, and more these probes are becoming available. They are really accurate. They can be reused, which is great. They're often easy to use. You turn them on, you put the probe in the water, and it gives you an answer. And they often do multi-parameters, so you can scroll through different parameters and look at. But they are really quite expensive, and they need to be calibrated frequently, which is a pain in the ass. But and it can really have effects if you try and take this into the field. You've got to take a whole suite of different calibration tools with you. The real advantage to probes now is the development more and more of automated reading systems, where you install a probe in your tank and it will automatically log data to your to a computer system, which you can access digitally. And in an area where you have access to Wi-Fi, etc., that can be really handy, and it can give you a real-time readout. And the systems are starting to become, relatively affordable in terms of hundreds of pounds rather than thousands. And finally, a spectrophotometer, which is kind of the gold standard. They're really accurate. They have almost no human error in reading, and importantly, they compensate for system watercolor. So they still rely on the principles that liquid reagent tests use, but a computer is able to see what color the water was originally and adjust for that. So they're much more accurate. But they're really expensive in comparison to any of the other tests. They need regular servicing about every year or two. And in many cases, they're probably more accurate than you really need. For a lot of parameters where you just are looking for, is there none of it, a little of it, a lot of it, they give you an answer to thousands of a milligram, which is kind of unnecessary. So when one is water testing, it's really important to monitor those particular parameters that we outlined earlier. So nitrogenous waste, pH, temperature, and some measure of alkalinity, hardness, or total dissolved solids. And not only is it important to know what you're testing for, it's important to decide on targets for a system based on the species biology and management knowledge, and also have some resources and plans in place to take action. But otherwise you end up in a system where someone is routinely measuring and writing down sublethally bad water quality and not able to do anything about it, or know that it's actually gone too far. So this is a system much like we use at ZSL, and essentially for a given system or a series of systems, we just have a key for all the different parameters and what targets are for the different systems, and what do if the values go outside certain levels. And this can be a really useful crib sheet to have to hand, and especially if you're working in a facility where there are lots of people involved who might be looking after animals. It allows some consistency between individuals and how systems are managed. There's no point going through all of this now, but it's still a useful system to think about. So we've looked at testing water, and now we need to think about where water comes from, because there are loads of different places you can get water from, and each of them have got their own pros and cons when it comes to amphibian keeping. So mains tap water is really, really easy. You turn a tap on in most places in the world and it comes on. And because it's treated to be safe for people to drink in most cases, it's also probably clear of pathogens because it's been chlorinated and treated in other ways. But the problem is that those same chemicals can be toxic to amphibians, chlorines and chloramines, and in some cases fluorines. There was a case in New Zealand where Archie's frogs were getting chronic metabolic bone disease as a result of fluorosis from the tap water, and also potential heavy metal contamination from pipes. And tap water varies both through time and space, and depending on what animals you're looking after, tap water may not always be suitable. So for example, in London, where I'm currently working, we have extremely hard alkaline tap water, whereas where I did my PhD in Manchester, the water is very soft and acidic. So still water coming out of a tap, but it's very different in each case. And also throughout the year, these can change depending on what's in the groundwater, how much it's been raining, and what, for example, farmers have been spraying on their fields. So tap water isn't necessarily always the same. Pond and stream water can be collected. If it's collected from the habitat of a species you're managing, it's obviously going to be relatively suitable if the habitat is intact. And then for animals that live on small food items like salamanders and their larvae, it's often teeming with food items for those animals of a really good diversity. And often it has bacteria in it which are useful for seeding your biological filter, which we'll come to shortly. The problem is it will also, as well as the food items of bacteria, potentially contain pathogens, parasites, and environmental contaminants from pollutants. So clearly we need to be careful of that, and we need to be especially careful if the target species has declined in this habitat. So if you rescue an animal from a disease epidemic, you don't want to bring the water from that place with you. And it may be really tough to transport enough water, particularly for large facilities, from a natural water source to your systems. And most importantly, we want to make sure that by taking water from a system, we don't desiccate that and put pressure on animals living there. Well or groundwater is again easy, usually it's a tap, but again it may contain pathogens, it's often not treated. It potentially can be contaminated with chemicals. I know at Jersey Zoo there have been issues with nitrogenous waste accumulation from runoff from agriculture into the groundwater, so very high levels of nitrates after it's rained. Because the water is compressed underground, as it comes to the surface, the change of pressure allows gases to come out of it, especially carbon dioxide, which can cause a rapid change in pH. Again, depending on what the geology is and what the time of year is, ground and well water can vary a lot. So it may not always be suitable for the species you're working with. Bottled water is really carefully controlled compositions. You know what's in it, and it's sterilized, which is great, but not with disinfectants. But using bottled water for anything other than tiny systems is expensive and environmentally unfriendly. There's a lot of variation between brands in the mineral contents, the pH, etc., and the content of bottled water won't necessarily be suitable for all species. We can use reverse osmosis to purify water, so that removes most contaminants, which is great, and the water is extremely soft, close to rainwater. For animals that live in collections of rainwater, like, for example, poison frog tadpoles, it may be more suitable than hard tap water. But its very purity can be an issue, and for animals to rely on more mineral content in water, this may cause osmotic issues and also mineral deficiencies. And the production of RO is necessarily wasteful of water. Sometimes up to sort of 95% of the water that goes into a unit may be discarded. So it can be quite environmentally unfriendly and expensive in that way. And finally, rainwater is free. It's suitable for species that live in rainwater. But it can contain pathogens, either from the rainwater. There's a little bit of data out there suggesting that some pathogens can be transferred in rainwater, and also for contamination with pathogens that may dwell in the receptacle that you collected in. There can be contamination with pollution from the air and from the roof surfaces that the water collects from. And again, you may have issues with not enough minerals for species that are adapted to high marine environments. And most importantly, what if it doesn't rain? You can't decide not to maintain your animals anymore because you've run out of rainwater. So from these lists, there's no one clear winner. And it's basically a case of looking at the different options, what's convenient, and what your species requires. But there are ways to address some of these problems. So we talked about heavy metal contamination and also things like chlorine and chloramine. There are chemicals available that chelate water and contaminants in water. So typical water conditioner basically causes these contaminants to drop out of solution where they're no longer biologically relevant. And you can also use carbon filtration, which I'll talk about a little bit shortly. If the gas content is unsuitable because of a pressure differential, so if you've used water out of groundwater or well water, just allowing that water to stand at sort of room pressure, potentially aerated for 24 hours, will allow gases to equilibrate between the water and the atmosphere, and then they'll become stable and suitable for use. If the pH is wrong, you can adjust this by buffering the alkalinity, for example, or by removing or adding leachates or acid sources. So things like sphagnum moss or tannin sources can suppress pH. And if the mineral content is wrong, you can use reverse osmosis filtration to remove all the minerals and then reverse dose it, so add in the amount of minerals that you want to get to the right level, either by adding salts or by just mixing back with tap water or similar. Or if you want to raise the amount of mineral, you can add sources of this to water or to your filter. So things like crushed limestone, crushed coral, and other components that will add minerals to the system. And in this way, we can manage some of those problems and get to a point where your water is suitable for your species. So now we've got a testing regime, we've got targets for the different parameters we want to maintain, and we've got a water source which is suitable and has been modified correctly. So now we need a system to put this water in. There are two main types of aquarium system that are worth thinking about. One is the open system. So in this picture, the blue box represents the tank, and you've essentially got fresh water, so clean water, continually adding in to the system and running out at the same rate. So it's relying on flushing to control the accumulation of waste products. So we don't need to worry about ammonia, et cetera, building up, because it's always being cleaned. And this is great because you don't get any accumulation of waste products to have to deal with. And the maintenance is relatively low. You don't need filtration equipment generally, and there's not very much else to do. You have to do water changes. Unfortunately, these systems require a stable and continuous source of water with appropriate parameters to work properly. So you need to have a lot of water to hand, because as soon as you stop pouring fresh water in and removing old water, there's nothing to stop any pollutants from accumulating. And if your fresh water source is compromised, you're going to be in trouble very quickly. It's also really difficult to manage biosecurity. Any amphibian collection should be considering the risk to the environment of escaping pathogens from the facility, even if they're not detected. And many of us manage those by treating with disinfectants. If you have lots of systems with fresh water constantly running out and waste coming out as well, it's very difficult to keep on top of storing, disinfecting and discharging all of that water. The other type of system is a semi-closed system. So this, rather than having water flowing through it continuously, uses a life support loop, which can be within or without the tank, to process things like nitrogenous waste, and then partial water changes to dilute the end product of it, the nitrate at the end of it. And this is beneficial because you don't have the reliance on continuous water flow, so you don't need to have an uninterrupted source of new water. And it's therefore much easier to manage both biosecurity, but also specialist water conditions. So if you're having to process your source water to make it suitable for the species you've got, you don't need to be continuously producing it. You just need to make enough to replace the partial water changes. The bad news is that it requires a lot more equipment and a lot more maintenance in order to keep it right. But in reality, the majority of us use this sort of semi-closed system to work. And you'll notice that there's open and semi-closed, but no closed system. And that's simply because in any aquarium, we are adding food, for example, which contains energy and especially nitrates, nitrogenous waste to the system. And that means at some point we're going to have to take out stuff later. So it's basically impossible to create a true closed system where you wouldn't have to throw any food in and you'd never have to take anything out. So one really, really useful and simple mantra to take away from this is that the solution to pollution is dilution. And this is where water changes come in. If you've got anything which is accumulated to dangerous levels in a tank, diluting it will always help the situation. So typically, based on water quality monitoring, you would do partial water changes of a system to remove the end products of the nitrogen cycle, and address any other accumulating toxins. And it's really important that the water that you put back into a tank when you've removed some of the old water is as similar as possible to the tank water in all the parameters, apart from the presence of waste products. So if your tank is running with a pH of 6.5 and an alkalinity of 10 milligrams per litre, the water you put back in should have similar parameters. And this is because we want to avoid large fluctuations in parameters in our system. So as well as using water the same as what goes back in, we want to do frequent small changes rather than big infrequent changes. So we essentially, within a semi-closed system, want to be as close to an open system as we can. And you can see in this graph kind of mocked up the difference. So if you've got any parameter on the y-axis and time on the x, you can see in the dark line, the solid line, that a parameter where water changes occur frequently deviates less from the original starting point than water which is left to be changed less frequently with larger changes, whereby a bigger peak and a larger crash happens. And those peaks and crashes can be stressful for animals and for the life support systems on which they rely. So we talked about filtration as part of an open system, and now I want to look at the different ways of managing this. There are 4 main types of filtration: mechanical, chemical, UVC, and biological. So I'm going to go through each of these in turn. Mechanical and chemical filtration are basically physical and molecular sieves, respectively. And all they do is collect contaminants in one way or another, either by catching it on a piece of sponge or similar, so physical or mechanical filtration, or by adhering it to a specific molecular matrix, so in chemical filtration. And this means that they've accumulated the stuff you want to remove, but they haven't removed it from the system. And so frequent cleaning and replacement of the media is really important. So mechanical media that you might use would include things like open pore sponges, where the water flows through a sponge and leaves the lumps behind, ceramics, canvas, gauzes and meshes, and there are a whole host of different things sold for different types of filter. But essentially they catch stuff. And chemical filters include activated carbon, and I'll look at again in a second, chemical exchange resins, so things like different resins you can buy which are specifically designed to absorb particular compounds. So for example, you can buy one that absorbs phosphate, another that absorbs ammonia. And it's important when you're deciding whether to use these to consider what your species needs and what your water source is. So if your water source is really high in phosphates, you might want to use a chemical medium that removes phosphates. So carbon filtration is, I'm just going to look at this in a little bit more detail just because it is so ubiquitous and it's so useful in so many different ways. It's essentially folded plates of carbon that form a series of fractal pores with charged surfaces. So different contaminant molecules that are charged get attracted in and lodge themselves against and fill up these pores. And it can remove all kinds of different things from the system, everything from chlorine, chloramines and fluorines from water treatment, heavy metals, pesticides and fertilizers. It can sequester dissolved organic carbon and tannins. It will remove growth inhibiting hormones expressed by, for example, anurin tadpoles, and also allelopathic chemicals produced by plants. And it will also remove antibiotics and other medication from the water. So it's important to think about that in the system. But this can therefore be a really useful thing to include in a filter because either for source water or within a system to help remove these things. But as for any chemical filtration, it will eventually become exhausted, and it's important to replace the medium as you go along. The frequency you have to do that will depend on the form of the thing and the context you've got it in. It usually comes in 2 different ways. One is just loose carbon pellets that can be placed in a filter, or sheets of treated material, so foam or gauze, which will act as a mechanical filter and also a chemical filter at the same time. So UV filtration is basically the use of ultraviolet C radiation, which destroys DNA and is therefore really good at sterilizing stuff. You essentially have a plastic tube which your water runs through, and within the tube is a quartz sleeve housing a UVC producing fluorescent lamp. Now the quartz sleeve is important because it allows UVC to pass through it, but not water, and that means that you don't electrocute your animals while you have a light essentially on in the tank. As the water flows through it, germs are exposed to the UVC and they are destroyed. And this is really handy because it creates therefore a low germ load environment, which is good for replicating extremely clean river and stream environments that many amphibians live in, or if you have a problem with a particular microbe, whether that's a pathogen or whether that's something which could cause environmental problems like an algal bloom. So this won't cure a problem, but it will reduce the number of organisms floating around in the water, which is a useful addition to any other management changes. And these essentially look like hose tails attached to some form of plastic hosing, and the light is housed within that. And you just plumb it into an external filter. So water goes in one hose tail and out the other back into the tank. So potentially the most important type of filtration you can use for aquatic amphibians is biological filtration. This basically uses cultures of bacteria, particularly Nitrosomus and Nitrobacter, to process nitrogenous waste. So you're using that, benefiting from that aspect of the nitrogen cycle where ammonia is converted from nitrate to nitrate by bacteria by culturing huge populations of bacteria in a filter through which the water is passed. So it feeds on it. And these bacteria are often a part of what is often termed tank slime. If you pick up any object from a mature aquarium, you'll find that it's kind of coated in a kind of sliminess, and that is a bacterial film, part of which will process waste. So it's important to protect that slime and not go overboard with cleaning. So any medium used for bacteria needs to have a high surface area because you want to have as many bacteria as possible able to grow on it. And suitable things are things like sponge, porous ceramics and inert rocks, sand, plastic beads, balls and wheels. And each of these different types of media is better for a particular type of filter. So once you decide the filtration you're going to use specifically for biological filtration, you can choose an appropriate medium. But bacteria are really, really sensitive, and it's important that we look after them because if you kill your bacteria, you lose the ability to process toxins in the water. So they are both sensitive to absolutely and change in pH. So extreme pHs that are arrived at over a long time will inhibit bacterial growth and metabolism, therefore the processing power, but sudden changes will kill them. Temperature, similar. They prefer warmer temperatures, sort of high 20s, but sudden changes will kill them, and extremes, especially extremely high temperatures, would also kill them. So we need to balance the temperatures that the bacteria need with the temperatures that the animals need, and especially cold water amphibians. We need to be careful that we're not overwhelming bacteria because they simply can't process as quickly at low temperatures. They are sensitive to light exposure, so it's important to avoid exposure to light, which can slow their metabolism due to shock or even kill them. They, in a way, feed off alkalinity, so especially carbonates in the water, so KH, in order as part of their metabolism. So the more minerals that are in your tank, generally, the more active a biological filter can be. And they also require oxygen, and they die extremely quickly if they're deprived thereof. And all these things mean that when we are, I don't know why these are doing that, that when we are cleaning filtration media, which you need to do to dislodge mechanically trapped dirt occasionally, we need to make sure we're doing that really carefully, as quickly as possible, while not exposing the bacteria to any of those parameters. And to make sure that we use water from the tank itself to clean the bacteria in, so that there are no sudden changes in any parameters that have been washed in the water that they've grown in. So where do these bacteria come from? There's two ways that they can get into your filter. One is they exist in sort of spore form in the air, and they will naturally colonize a system unless it's kept hermetically sealed. Or you can inoculate from an existing mature system with similar parameters. We need to be careful of doing the latter because, as well as introducing beneficial bacteria, we may also introduce pathogens. Once you've seeded your enclosure, it's important to make sure that your tank is fully cycled before adding animals, because at first there will not be enough bacteria to process a heavy load of ammonia from the amphibians. So what we can do is set the tank up and start to feed it, either with ammonia itself, which can be added at a suitable concentration, or with something that rots and therefore releases ammonia. So people often use fish food or similar. And if you measure throughout that process the levels of a nitrogenous waste, what you'll see is peaks in ammonia, so the red line, then nitrite as the ammonia goes down, and then nitrate as each stage in the nitrogen cycle establishes and begins to feed and grow. And eventually you will end up with almost undetectable levels of ammonia and nitrite, and slowly raising levels of nitrate that you can manage mainly by water changing. Now, there's a scale on the x-axis of 10, 20, 30, 40, 50 days. This is highly dependent on temperature. At a sort of tropical temperature in the high 20s, you can expect filter maturation within weeks. At very cool temperatures, so for example, for temperate salamanders, it may take months for a filter to fully mature. So it's really important to monitor not only this maturation process, but what these levels of things are doing after you add animals, and make sure that you're adjusting settings in response to your tests. So don't assume that a filter is mature just because it's been there for long enough. Now, as well as bacteria, plants are an often overlooked aspect of biological filter. They use nitrogen for growth, whether we're talking about higher plants, tree plants, or algae as well. And they also take up nitrogenous waste in a preference order. So we often think about them taking up nitrate, but they actually prefer to feed on ammonia. If ammonia is not available, then they'll feed on nitrite. If not that, they'll go to nitrate. And that means that they uptake the most toxic waste first. They also will sequester heavy metals, especially fast-growing plants. And therefore, removing plant matter occasionally from an aquarium actually removes nitrogenous waste, and they can be a really useful backup to biological filtration with bacteria. If you can grow plants like this peace lily's been planted with the roots in the water growing hydroponically and the leaves above it, they will process ammonia, nitrogenous waste faster because they have more access to carbon dioxide. And you can also use algae. So this top picture is from an algal scrubber where water is poured over an algal culture which is brightly illuminated before re-entering the tank. And these are extremely good at uptaking nitrogenous waste, contaminants, but also things like heavy metals and phosphates that the plants need to grow. There's a really good book called The Ecology of the Planted Aquarium by Woolstead that is available online, and I'd recommend it if you want to look at plants for managing water quality, because it's a really good treatise on that. It's also a good idea to remove uneaten food, for example, or dead animals before they contribute to the ammonia level in an aquarium. So sometimes we can add small invertebrates that will eat organic matter before it decomposes. Things like shrimps, isopods, and snails. These are also handy because they can also become a food source for the inhabitants of the aquarium. But we need to be careful because some of these carry diseases on their own. So for example, crayfish and other decapods may be transmitters of BD. So I'm now going to just look at a couple of types of media for filtration for biological filtration. There are hundreds of different sorts, so it's not worth going through all of them here, but they fall into two main sorts. First of all, water moving through static media. So this is the majority of filters with which you might be familiar. So a pump forces water through fixed media or a block of, or a layer of media. And these are really efficient. These can be a bit less efficient, but they're really easy to use. And because the medium is fixed, they survive for longer without water flow. So if your pump fails, there'll be a bit of oxygen dissolved in there, which they can use. But different parts of the medium will receive different amounts of water flow, and therefore they're not fully efficient. The other way round, where water is moved through moving medium, is a bit more efficient because every particle in that filter will be equally exposed to the water column. And these are things like bubble bead or super bead filters that you have for managing large systems, fluidized and non-fluidized bed sand filters, where sand is tumbled around in the water and the bacteria grow on the sand, and coldness reactors, which are similar but using plastic sort of wheels. The problem with these is as soon as water flow stops, all the particles settle out of the water column and exclude oxygen, so your filter will die extremely quickly if it fails. So how do you choose the appropriate filtration? There's no one appropriate solution, but it's important to think about the type of habitat you're trying to recreate. So are you going for a crystal clear mountain stream or a kind of muddy ditch? How big the system is and what the stocking density will be on it. So how much water do you physically need to move, and what sort of loads of biological waste you need to deal with? And also what can you afford? It's really important though to build in redundancies and to slightly overspec a system in case of a problem. So you want to allow for a piece of food not being removed and rotting down, or an animal dying and not being detected, and you want the system to be able to deal with that rather than crash. So for example, in these tanks, which were housing some salamanders, you have external canister filters. There's one you can just see at the bottom left of the picture. But there are also some in-tank sponge filters, which means that if one fails, there's at least some capacity to process waste left there. And another really important point to make is that if you can't meet the requirements for a species, it's best not to house that species rather than make do with something which is suboptimal. So I mentioned stocking density. The more volume, the more animals you have per volume water, the higher the nitrogenous waste load there will be, the higher the germ load, especially if animals become unwell, and the higher the concentration of things like growth inhibitors that will affect the development of tadpoles. So the capacity of the system and the maintenance routine that's designed should be made sure to cater to stocking density. And these things need to be planned in advance, especially because animals will affect density as they grow. So it's worth thinking about the total mass of animals rather than the number of individuals, because the newly hatched tadpoles produce a lot less waste than animals that have reached their maximal size. So how do we control temperature? Heating is really easy. There's a whole bunch of different equipment available for doing that. Cooling is a bit more difficult and a bit more expensive often, but you can get things like chillers or air conditioning for the room at the high end, or you can literally, for the short term, if you have a particularly hot day, add frozen water bottles to your aquarium to drop the temperature. But overheating is a really difficult thing to deal with chronically, and it's really important to make sure that you properly design a system and housing for a system so you can maintain proper temperatures, especially in the height of summer. All of these things require water flow to either work at all or to prevent accumulation of sort of temperature gradients within an enclosure. So how do we put all these components together? So imagine this is your water column, and on the left is the starting point when your water goes into your life support system, or LSS. And you've got a whole bunch of different components. You've got large and small, coarse and fine mechanical waste, ammonia, nitrogenous waste. You have chemicals that you need to remove, like chlorine, and then you've got bacteria and other organisms that need to go. So you first of all start with your coarse filter, and that allows everything through other than the really big lumps. And that's good because that means that when you go through your fine filter, the big lumps won't block it up too quickly. That leaves you with just your nitrogenous waste, undesired chemicals, and any microorganisms that you want to remove from the system. So by running them through then your biological filter, you've stripped out toxic ammonia and nitrites, and then your chemical resins go even after that to avoid them being blocked up by mechanical waste, and that just leaves your bacteria that you want to get rid of, and algae. Now, because you've removed all of the mechanical waste from that system, they have nowhere to hide, so they're zapped by the UVC. And finally, you have any inline heating and cooling devices, which you don't want to get clogged up with waste. So that order of different media is really important to maintain. Otherwise, you'll end up ruining some media and reducing the efficacy of others. So we've talked about lots of different terms and parameters, etc. And while it's important to think about each one in turn, it's important to think about how they relate to each other. And I'm not going to go through this in any detail, but you can see from this diagram how loads of different parameters affect one another. And we need to remember that if we change one parameter, so we change the temperature, we will change lots of other things in the system, and we need to be prepared for what that will do. One really important interrelationship is that between ammonia, pH, and temperature. So nitrogenous waste that's in the ammonia form can exist as one of two types of ion, ammonia and ammonium. Ammonia is way more toxic than ammonium, and these two things will be in dynamic equilibrium. And the proportion that is in this highly toxic form depends on water temperature and on pH. So the higher the temperature and the higher the pH, the more toxic a given amount of nitrogenous waste is. And this may all seem academic until you put it into the context of a really simple thing of unpacking some tadpoles from some bags they've been transported in. So this sealed bag with the tadpole arrives, it's been in there, and it's been in there using oxygen. So it's been expressing CO2 into the water, and it's also been releasing ammonia as waste. Now the CO2 that's dissolving in the system is going to suppress the pH because it becomes carboxylic acid. And because the pH is suppressed, the dynamic equilibrium between ammonia and ammonium is pushed towards the less toxic ammonium, so the animal's fine. We then cut the top off the bag, that changes the pressure, and all that CO2 that's accumulated in the water gases off. The pH then goes up because it's less acidic, and the dynamic equilibrium shifts so that ammonia now dominates. And our tadpole then dies. So this is a really, really simple but important example of how the interrelatedness of parameters needs to be considered when you do anything with a water system. So just finally, I want to go through the 10 commandments of water quality, which are to start with the right sort of water. Thou shalt maintain good water quality through appropriate combination of frequent partial water changes and proper flow through and filtration. To make sure that media are cleaned and replaced properly, to look after your bacteria, not to overcrowd or overfeed animals, and to remove uneaten food as quickly as possible. To incorporate live plants at all possible into a system, to remember that parameters are interconnected, to remember that fluctuation is bad for everything, to make sure that you know your species, use data from the field and from colleagues as much as possible to inform how you maintain the water quality, and to make sure that you keep regular records and share information so that everyone can work together and benefit from it.